Thank you all for coming and for listening to me. I'm CEO of FiveAlarmBio, and I'm going to tell you a story of how the company has evolved over the past 2 years, how it started and how we evolved. So if I could go to the next slide. We're actually working in aging, and if you could think of how old you will be in 2040, you'll make you realize just we are all aging, and this is going to impact all of us. And actually I see in the really long term that we'll have molecules, and hopefully some FiveAlarmBio molecules in the clinic, in the market, that we'll be taking like we take statins today, that will keep us healthy, prevent chronic disease in the long term. But our companies can't go straight to that product. So we are developing products for chronic diseases. And that's our main goal right now, is to get evidence of efficacy in chronic diseases. So this company was founded on the science from William Baines. He's an absolutely brilliant scientist. He's very creative and he's an entrepreneur. And he has been thinking and working and excited by the biology of aging since the mid-2000s. And he was at a conference and somebody presented some data that was on a compound that was in the clinic. And he saw that data and he recognized that that compound could be really useful in an aspect of aging that he was really interested in, that he could use that compound to probe his interest in aging. So we've called it our probe molecule, and it's Fab1. This is not the molecule we want to take to market, but it's the molecule that's the basis of our mechanism. And so he talked to Sunil, he talked to Anne Baker, strategic consultant in pharmaceuticals, and they all decided to put some money in. They enabled William to do some early research to prove whether this molecule truly had efficacy in some aspect of aging, in his mechanism. I can't disclose the mechanism, so I feel a bit vague. And he did some cell culture work, and he showed that you could increase the longevity of cells, primary cells, human cells in culture by 40%. And that was remarkable and surprising. Even William couldn't believe how good it was. They talked to David Vinenkove. He is a key opinion leader in aging, and he is an expert in models using C. elegans, and that people recognize that C. elegans is a great early model for aging. And he tested that compound Fab1 in model of aging in C. elegans, and it increased the healthy lifespan of C. elegans remarkably, and by 40%. And he said that that was the best molecule they had ever seen at that point in his experiments. So really interesting. Company had been formed by the 3 people, but they hadn't really got it operational other than William in the lab. So I joined the company. We needed to, William he applied for an Innovate UK grant. He got that grant, 360,000. You need money to run the grant. You need scientists to do the grant. So I joined the company to help raise the seed round, which is £500,000. So with £500,000 and the Innovate UK grant, we've been working for 2 years to get more evidence in this compound. We've got fantastic scientists. Hannah Mizzen, she's from the lab, Professor Chloe Bishop at Queen Mary. And we've got Peter Tyrer, a very experienced assay biologist. And these 2 people have been doing most of the lab work at Baby Home Research Labs. And they've got remarkable data. So if I could go to the next slide. So we now know that we can develop small molecules to modulate the key components of the cell's damage repair pathway. Based on Fab1, O2H made a lot of chemical compounds for us, and we now have 100-fold better potency than Fab1. Fab1 we know is safe in the clinic, so we think this whole mechanism is a safe mechanism to go for. We've got the chemistry. We've established a development pathway route to clinical efficacy. We've actually demonstrated functional efficacy now. We think these are all significant advantages for this stage of company. So if you go to the next slide. So this is the data on over here, just to show you some data from William, which was the C. elegans data showing that Fab1 increased the healthy lifespan of C. elegans by 40%, and which is stated without showing the data on the healthy growing time of cells. So the next slide. So this is some efficacy data that we've shown now. These are in simple models, okay? But we're a small company with limited funds where we started, and we just wanted to show that it actually could be useful in some models of disease. So over here we can show the cell migration scratch test, the grey cells, primary human cells grown on a surface of a plate. If you put a scratch across the surface of the plate, the grey cells then migrate to fill that scratch or that cut. And their migration rate is increased significantly in the presence of our compounds. And these are not easy things. They're quite remarkable. This data is really unusual. So Fab15 is really half the time taken to wound closure with Fab15. This one on the left is the post-operative delirium. This is in collaboration with the university. We want external scientists to validate and to be interested in our science and show that we're not alone in thinking these. This is a really interesting aspect. So we tested in zebrafish, a model of zebrafish growing healthily, and it anaesthetized them, and we measured the recovery of the zebrafish from anesthesia. And it halves the time taken for recovery from anesthesia, which is amazing. We think that this might lead them to be able to be tested in the efficacy in hopefully a clinical trial in the longer term for post-operative delirium, which is a massive issue in the clinic when older people have orthopedic surgery, often end up in intensive care. So these are just simple models, but they show that our mechanism really can be intervened. It's not pipe dream that this is real. So if I could go to the next slide. The other thing we've done is worked with Pelargo, and we have a putative target working with a Pelargo-Setzer study. And with we've got the target fits in with Williams' hypothesis and the biology we see under the microscope with our cells. And we have come up with reasons why we think that these molecules will work in different indications. So we think it work in wound healing. The scratch assay shows that's possible. The post-operative delirium, the anesthesia assay shows is possibility. We think it will actually work in slowing down the onset of Alzheimer's disease, Parkinson's disease, and muscle wasting sarcopenia. So a number of different diseases, and we can explain why we think that if we get CDA and can talk about our mechanism. So these are all things we want to test out. So if you go to the next slide, we're raising, so obviously we want to use the funds. Our next fundraise will be IP, composition of matter is really important. And we have some reviews that say we have some of our compounds are definitely patentable. Next slide. An interesting comments made earlier about exits and value and who your customers are. We want to exit with clinical efficacy in at least one indication, and we could continue to develop in different indications, but we want to license the first molecules. And so our customers will be the pharma companies in the first indication instance. And Lilly have given us a letter of support already. They're following us. We know other pharmaceutical companies are interested. They're talking with us. So they're very interested to know whether this mechanism really will pan out in the longer term. So the next slide. We are raising £4 million of funding in order to get to our first molecule in a first indication to the clinic to prove efficacy, and that this pathway really can intervene in disease. So we're going to use the money for chemistry, for biology, to show that this can actually have opportunity in different disease indications, and to get to pre-IND with our first drug molecule for potentially at the moment for wound healing, because that's a low-cost, fast route to the clinic for proof of concept. And I've already talked about the people that are involved in the team, and you'll see in the next slide, we also have significant key opinion leaders as well, including our own Andy Morley here from Motivate. So thank you very much for your attention.
Mark, then Mark. And then...
What's your clinical validation? There are lots of compounds that extend lifetime of cells and elegans and apart from maybe metformin, none of them are translated. So if you look at human biology or conventional pharmacology in humans, what confidence do you have your target is meaningful in humans?
Yeah, well, we see the target being meaningful because it ties together with our understanding of the pathway, and we see those changes in cells, and they could be converted into biomarkers so that when we get to the clinic with our clinical samples, we can measure them.
Gain of function, SNPs, human genetics.
Yeah.
Association of target with disease.
We see a reduction of senescence. We see a reduction of an increase in mitochondrial function, and a number of other factors that we actually measure in cells. And all of those, the things that we see in cell culture, are translatable into the clinic.
Thank you. Mark?
Just a quick one. I mean, I think very interesting and love to know more. And obviously, I haven't signed the CDA, but I mean, you know, it's all about cell fate at the end of the day, and we're programmed to die as a species for obvious reasons. And I think, you know, doing at Revolver is we're tapping into transcription factors to, you know, accelerate that death where, you know, for example, development goes wrong. Obviously, you can't tell us, but what is the fundamental concept behind the company? You know, how are you going to, because the problem is you have lots of small molecules, you'll need to actually interact with multiple pathways. It's very unlikely you'll get a node point unless you're going to something right at the top.
Yeah.
You know, so Yamanaka factors, you know, modulate transcription factors and reprogram you to being, you know, where you were.
Yeah.
What is the actual philosophy behind the company? Because I thought it's great, you've got all this data, but what's the thinking behind it?
So it's a protective mechanism. It is central.
Yeah.
And it does feed impact on all the different markers of aging.
Yeah.
And we see that in the cell cultures. So if we do limiting dilution assays for cell senescence and aging, and we can see the impact this has on all the different aspects of aging. So it's protective, which is why I think in the very long term, when we've done huge studies of people, I think it will be beneficial just generally. But in the meantime, I think it enables our bodies to be more youthful and respond properly to chemical damage, to any damage that's happening, and enable you to heal yourself in the end. Yeah.
Thank you. All right.